Okay, good afternoon and welcome. I think we'll get started. Waiting for a few more stragglers to come in after lunch. But I think this is going to be one of the most exciting panels ever to be presented at NIBA. The speakers on this panel have been involved with the development of some of the most successful pharmaceutical products ever developed. And we'll hear about the trials and tribulations of such blockbusters as Mevacor and Feldene. And I'm sure I'm not going to spend too much time up here talking so that we can get to the exciting part. So the first speaker is Dr. Joseph Lombardino from Pfizer. Dr. Lombardino is presently Senior Director of Operations Planning of Pfizer Central Research in Groton, Connecticut. He has spent his entire 40-year scientific career with Pfizer Central, and for 29 of those years, he was a medicinal chemist designing and synthesizing new chemical compounds as potential therapeutic agents. In 1976, he established and headed the Department of Development Planning at Pfizer Central Research, helping to bring a matrix of teams and project management techniques to the drug development process at Pfizer. He received a PhD in organic chemistry from the Polytechnic University of New York and an advanced diploma in chemical sciences from the University of East Anglia in Norwich, England. He holds 56 U.S. patents on various medicinal agents, including the patents covering Feldene, a novel anti-inflammatory agent and a major therapeutic agent worldwide for the treatment of arthritis and related diseases. Dr. Lombardino is a member of the American Chemical Society, the International Society of Heterocyclic Chemistry, and the Inflammation Research Association. Dr. Lombardino was named the 1989 Eli Whitney Award recipient by the Connecticut Patent Law Association for significant contributions to law or science, and he was also awarded the 1989 Distinguished Alumnus Award by the Polytechnic University of New York. So with that distinguished introduction, I'd like to pass it along to Dr. Lombardino.
Thanks very much, Pat. Okay. Forgot what I was going to say. My personal involvement in the story of Feldene started 35 years ago. So my presentation will describe how a major drug was brought from the lab to the market in those days. Nevertheless, I think there are lessons to be learned from this experience that are relevant even today. After I briefly describe the process by which generally a new drug moves from lab to market, I'll describe the Feldene story for you, and then contrast the Feldene story with pharmaceutical R&D as it is practiced today. The medicines we have today, I can categorize as having come from three major sources: from folklore, from accidental discoveries, and from the pharmaceutical research process. And some examples from each of those categories are shown on this slide. As you can see, pharmaceutical research is by far the largest source of our modern-day medicines. These medicines are not accidental discoveries, generally do not come from natural sources like morphine or digitalis or quinine did, but for the most part are designed and synthesized new chemicals that are intended to target a specific disease. On that list under pharmaceutical research, you will hear today about Feldene and Mevacor and Zocor and how they came to be major drugs. First, to the general process for finding new drugs. The long process needed to produce a new drug has a number of discrete steps, which I'll overview in 3 slides. If we look at the first one here, the process begins here when management at the company decides to invest in a new research area. People are hired, laboratories are assigned, equipment is purchased if needed, and this team of people sets their objectives. They need to evaluate what's out there now, what are the deficiencies in the present therapies, and what they believe they can do for finding new and effective new therapies. Once their objectives are set, then begins the process of either synthesizing new compounds or high-throughput screening of existing compounds or fermentation of new compounds, or any of those processes used to discover a lead compound. And I can tell you from my personal experience that these compounds that are synthesized and designed with a specific target in mind, all are tested, and about 90 to 95% of them fail in that first step. They just do not exhibit the kind of activity that's desired, the level of efficacy, or meet the other criteria. But that negative information that we generate most of the time is fed back to the laboratory to enable the design and synthesis of yet additional compounds. And so in a cycle, with the objective set, lead compounds being synthesized, tested, generally found to be negative, and the information being fed in a cycle of that type. Eventually, one compound is shown to be active in the tests, the initial tests, and that's designated a lead structure. With that lead structure in hand, the chemist can then prepare related compounds, so-called analogs, in order to improve the efficacy and minimize any side effects that the initial lead had. So here again, we're doing a cycle of synthesis and learning about the structure, activity, relationships in this lead series. From that work, generally will come one compound that meets the criteria, at least initially, and that is what is designated the drug development candidate. So we're moving from the R, the research part, into the development part, the D of R&D. That drug candidate then has a series of hurdles that it must overcome. Formulation research, stability testing, scale-up synthesis, and chronic safety in animals. Most of the time, that lead compound, that drug development candidate, will fail in one of these tests. And once again, that negative information is fed back to the team so that additional structures are made and tested and brought into the development process. Eventually, we come to our first pile of paper in this process, the so-called IND. We have a compound that has passed these tests, passed these pharmacological evaluations, and we're asking the Food and Drug Administration to allow us to test that new compound in man. And the summarization of all of our work is in this pile of paper called the IND. On this slide, we're moving from right to left in the clinical process as we go from phase 1 in normal volunteers, where the new compound is studied for its toleration and pharmacokinetic properties. And a number of these candidates fail here because despite all the safety evaluation that's been done in animal models, man is not a monkey and is not a rat. And too often we find toxicity in man at this stage that again would cause us to cycle back into the chemistry laboratory. But for those development candidates that pass to phase 1, we then move into phase 2. Here, the compound is tested for the first time in those patients for which it was designed. And the simple question we ask here is, does it work? Is it efficacious? Does it do anything for these patients? Unfortunately, too often, and in fact more than half the time, the answer is no, it doesn't do enough, or in the patients, some side effects are showing up that would cause us to reject this candidate. And it's back to the laboratory again. But for those clinical candidates that pass this phase, we move now into the largest, most expensive, most time-consuming phase of the drug development process, the so-called phase 3 clinical trials. Here we're studying the new compound in thousands of patients around the world so as to build a large safety and efficacy database from these patients. When that's completed, and I should add that in our experience, most of the compounds, not all, but most survive that phase 3 testing. When that's completed successfully, we come to our second and largest pile of paper, the NDA, the new drug application. In this document is summarized all of the research that's been carried out to date, everything from the chemistry laboratory experience, the synthesis, the manufacturing details, the toxicology, and all the clinical trials, every patient reported in that document. And the objective is to have the experts, the corresponding experts at the Food and Drug Administration examine that dossier and decide whether the research we've done is accurate and sufficient to support our claims and the safety and efficacy that we're claiming. Only with the Food and Drug Administration's approval can we then move to the marketing phase, bringing the new drug, the new medicinal agent, to the patient population for which it was originally designed. Now, for those of you that have been summarizing the time parameters that I've shown on this in the previous 2 slides, you can see that it takes 10 to 20 years to accomplish this for a single new agent. And the cost figures for this range now between $400 and $500 million for a single new agent. In fact, if you look at the development timeline with the specific activities as shown here on this Gantt chart, from the time the compound enters development, let's say a new compound is entering development this year, we call that document a recommendation for development. With management's approval to take that new compound into development, it must pass all of these tests, as I mentioned, phases 1, 2, and 3, carcinogenetic studies, filing the NDA, waiting for approval. The compound entering development today, if all goes well, will enter the marketplace in 2005. Unfortunately, as I mentioned already, things do not go smoothly. A company like Pfizer synthesizes worldwide about 8,000 to 10,000 compounds a year. Every one of those is designed and synthesized to be a potential new drug. In an average year, we put about 18 compounds into development. And of those 18 that entered development, at the other end of the pipeline, a decade later or so, will come one successful candidate. That's a very, very low batting average for 10,000 to get one out the other end. And I've already mentioned the cost and the time parameters involved. Now, the cost for doing this work has been escalating over the years. Here's some data from the literature starting back in the 50s, where one could carry out a complete development program and file an NDA and get a drug on the market for somewhere less than $10 million. That's been escalating, the cost of that development program, such that today it's well over $400 million on our own experience at Pfizer. And the reason for that is the complexity of the program has increased, the regulatory requirements have increased. All of these things combine to require larger programs with more data and still larger NDAs. I don't know what that is. Oh, I know what it's supposed to be, but I don't know why it's black. Somebody have vacation slides they could show? Okay. I tried to convince you of the lengthy, expensive, risky R&D process. I hope it succeeded in that last segment of my talk. And I want to turn now to a real case history, the discovery and development of Feldene. The Feldene story started in 1962 with a management decision at Pfizer to begin anti-inflammatory research for the first time. A team of 2 people, a chemist, that was me, and a biologist, were assigned the task of finding improved therapy in an area that Pfizer had never before researched. That is the treatment of arthritis and other painful inflammatory diseases. We had to become experts in the field as quickly as possible. Oh boy. To assess the literature, the relevant patents of the day, and then over the next 6 to 12 months or so, we developed a set of goals for ourselves, and here they are. We decided that what we would seek would be a completely novel structure, that the compound would be an acidic compound, but would not be from the class known as the carboxylic acids. We decided that the acidity that we wanted would be in this range of 4 to 7, that the compound should have a long plasma half-life in vivo, and that the compound would be very potent as we moved from rat models to dog models and into man, and that it had to be active by the oral route. And finally, safety was a very high criteria for us. All of these agents need to be safe, but we felt and recognized that this would be an agent for the chronic treatment of arthritis, chronic administration over long periods of time, so safety was of primary importance. And now I'm going to try to change the slide. Yay. But it's backwards. But it's backwards. Well, you can't have everything. I do, but you don't. Well, we began our chemistry work after the objectives were set, and that chemistry effort produced 4 classes of active agents over the next 5 years of work. Each of the series, which I hope you will see in a moment, was designed and synthesized and then tested. Multiple, sometimes hundreds of analogous structures were synthesized and tested in each of these classes. Each of the series had some desirable biological activity that was of interest to us, but none ever produced a clinical candidate. Next one. So from these 4 families of compounds that we worked in were days of great excitement. They were working in the animal models, but none of these ever progressed to testing in the clinic. After 5 years of research, we finally got into a novel series of acidic agents, later to be called the oxycams, the last 2 shown on this slide. These agents, at least the first few that we made, began to meet our goals and were surviving in the animal toxicology. The earliest oxycams that we took into clinical trials for treating arthritis was one coded CP14304, which when taken to man was found to be too weak as an anti-arthritic agent to meet our goals. A second analog was taken out, a compound called pseudoxycam, which was highly effective, but began to show liver function test elevations, which we thought possibly were drug-related. At that point, we began to worry about whether this whole structural class in this class, could we ever combine adequate safety and potency that we were looking for in clinical trials. Our management was split over whether to continue to fund this kind of research. Fortunately, at the time, I had piroxicam ready with all the preclinical testing completed, so they decided to take one more oxycam candidate out to clinical trials. With that decision, Piroxicam was eventually shown over the next few years of testing in man to have the sought-after potency and the safety, and it eventually passed all of the phase 3 clinical trials. All the knowledge and experience we had gained over 5 years of failure of working in this area of anti-inflammatory drug research had paid off. All of the goals we had set for ourselves were fulfilled as shown on this slide. This, at the time, was a structurally novel series, never before known in the chemistry literature. It had the appropriate acidity that we were looking for. It had the long plasma half-life, both in animals and later in man, such that a single daily dose was sufficient to treat arthritis in humans. And it had the high potency. At the time, it was the highest non-steroidal, highest potency non-steroidal, where a single 20 milligram dose was sufficient once a day to treat arthritis. As I mentioned earlier in my talk about the process, the NDA is the document that summarizes all of our research for review by experts at the FDA. I thought you might be interested in seeing a picture of the Feldene NDA. This NDA was assembled into 200 volumes, 60,000 pages long, which was big for those days, and was sent to the FDA and other regulatory agencies around the world. And as I mentioned earlier, summarized in this document, that is this pile, is all the preclinical, all of the clinical, all the manufacturing data, et cetera, all the case report forms for every patient that had received the drug. Regulatory approval in the US and overseas began with filings in 1978 and first approvals in Europe at the end of 1979, and then in other major countries overseas in 1980. The review at the of the US NDA dragged out for 2 more years before approval came in 1982. So here's the timeline for Feldene. From the idea in 1962 through the development process that started in 72, through the filing of the NDA in 78, and the approval of the NDA in 82, where it finally reached the marketplace in the US in 82, took 20 years. A very long time, a very expensive process, but in the end it produced a very valuable new drug. Piroxicam went on to become a major therapy worldwide for the treatment of arthritis and a major commercial success for Pfizer. Now today, there have been a number of changes in this process since those days that I was describing to you. There are many time-saving techniques that are being utilized today. I list a few here. The robotics and the high-speed testing techniques that are utilized that speed up the testing of the new chemical compounds. The high-speed chemical synthesis that's used to follow up the lead compound so that many more analogs can be produced much faster. The use of human receptors is improving the drug specificity. Once we find the lead by using human receptors, we're getting much more targeted and specific activity. The use of the computer has definitely assisted in this process in its ability to store information and for the chemist and the biologist to retrieve that information in a very facile way. And finally, I should mention that the review time at the FDA has shortened due to the Drug User Fee Act. All these things combine to make the process better today than it was in those days. To close, I'd like to say that these are all major changes that are speeding the process for moving a drug from an idea to the marketplace. But what is unchanged is the innovative, creative thinking of the scientists involved. The medicinal chemist is still designing molecules and investigating the structure-activity relationships in order to maximize efficacy and pharmacokinetic characteristics and to minimize side effects. The biologist is still creatively testing the new molecules in appropriate animal models. And there are dozens and dozens of other highly trained specialists involved in the process, working in a team environment. Progress has indeed been made. and the future looks bright for finding these new therapies. But the vital role of the innovative scientist in this process remains unchanged. And there's still much work to be done, as shown on this slide. Pfizer has teams of scientists conducting research in each of these disease areas. With the financial support coming from the pharmaceutical industry and the dedication of the highly skilled scientists, I'm confident we'll see important new therapies for some of these diseases in our lifetime. And these therapies will come from this pharmaceutical research process. Thank you. Are there any questions? Yes?
Just out of curiosity, and excuse my naivete, but after 20 years and all these dollars in R&D, about how long does it take to recoup on your investment in such a drug?
Well, let me say two things. For those drugs that make it to the marketplace as a group, generally only 1/3 of them will recoup the investment, okay? So if you look at Feldene, it's an exception. Yeah, the company recouped its investment within that first year. There's no question about it. But the income that comes from that success is helping to support all the failures that I told you about in many other projects and within our own project. So that revenue stream is what feeds the research process to assure that those few successes that come out will bring us the novel and important new therapies that we need. We set that up, which was discovered at Merck, which is why Al is laughing over there. And we adapted that model to our environment and set it up and automated it in such a way that we were able to screen a lot of compounds very rapidly through there. And that model turned out to be quite predictive as far as potency from the rat to man.
What was the starting point of the chemistry?
The chemistry, as I indicated in the objectives, was not to look at carboxylic acids, but to achieve the acidity we wanted. So enolic acids was where we started. And as the first ones we found were the indane diones, which, as I say, were weakly active. We never got a candidate. But that line of thinking was promoted throughout the next 5 years, leading to the enolic acids, the oxycams. Yes?
Was there a point in time in the beginning before you began to get success that the company had decisions to make as whether to continue or not? Or was
No.
it support all the way through?
No, in those early days, when the company, this is generally true, when the company makes a decision, a large company, they hang in there for a few years, giving the team enough time to get their feet on the ground and to begin to find things. I don't know what would have happened if we had nothing to show after 3 or 4 years, but certainly for the first few years, we didn't have very much, they didn't bother us very much. Because by the time we had our 4th failure, then as I mentioned, we were being questioned about whether we should stay in this area or not, and whether we should be looking at carboxylic acids, which we didn't.
Even when compounds are killed, they sometimes have a miraculous life and reappear because the scientists are still driving for it and find a new potential direction in the larger community.
That's right. We have a bank of compounds and it's over 500,000 in number. About 5,000 of those are ones that I've made over my career. And they're in there, and people draw upon those now via the computer. They can search for structures, they can search for physical characteristics, and test those. A number of mine have shown up in other tests quite unexpectedly. No other questions? Thank you.